In this lesson today, we're going to have a look at comparative DNA. So before we can really talk about comparative DNA, we need to talk about how we can get these DNA sequences themselves. So how do we do this? Well, we use a process called DNA sequencing. And what this allows us to do is to determine the order of the nucleotides or bases in a particular gene that we might be sampling, or a small segment of DNA itself. So DNA sequencing has been really useful for us as humans to not only map out our genome, so the human genome, but also of other species' genomes. It's allowed us to identify different diseases and mutations that have arisen from the genetic code. And the focus for us this year is that it's allowed us to identify some of the evidence to suggest this theory of evolution. So two particular parts of this DNA sequencing process. Number one is PCR. So we've spoken about PCR previously, and there's a recording of that lesson. PCR is just the process that we use to amplify DNA. And then secondly, we can use gel electrophoresis, and as a component of that, we're going to separate those DNA fragments by size. So when we're talking about the DNA sequencing and using the gel electrophoresis machine itself, it's really important that firstly we've identified we're separating the DNA fragments by length, and they're going to spread out across the gel bed. But what we can then use is electronic devices and computers with a laser to help detect a particular tag or marker that we might introduce. Now, this is commonly used in what we call the Sanger technique, and we'll explore that on the next slide. So when that particular marker, that particular tag reaches the end of the gel, the laser detects it, and this provides us with the particular sequence. So when we have a look here, this is a visual breakdown of this Sanger technique. So we can see the deoxynucleotides up the top left-hand corner. These are like our fluorescent markers, and you'll notice that they occur at the end of each DNA sequence. So for instance, when we're looking at guanine in yellow, so I'll circle it with an orange box here, we can see that this is the marker at the end of that genetic sequence. We can then see as we're taking that out and placing this particular fluid, deoxynucleotides, all into our gel and it separates out, we get the three different bases for guanine being detected, and we can detect their frequency and correlate that to their length as well. So I'm just going to skip through some of this now and talk about the ethical considerations for us. Four main ones, really important to be able to describe these. So firstly, autonomy, which is the right to determine if a particular individual wants testing to be completed. This is different from confidentiality, where the use of the genetic material once it's been tested, that's sensitive, right? So keeping it confidential. Number three, equity, so equal treatment regardless of genetic information, so ensuring everyone has the same outcomes. And privacy as well. Okay, so that people themselves have the right to their results independent of others. So when we then talk about comparative genomics, okay, we're now talking about how, although while we all have DNA and every species, living species on Earth has DNA, the sequence of the bases varies. Okay. So we've spoken about speciation previously, where a new species would have very similar DNA to the original species. But over a long period of time, what we find is that new species gradually accumulate more differences. And that's due to a combination of, firstly, mutation, secondly, natural selection, and third, genetic drift, all of which we've spoken about previously. Now, the final definition that's really important here that we need to make sure that we include is this term here, genome. So a genome, very simply, is just that complete set of DNA found in every single cell of a particular organism. So when we look down the bottom of this slide here, we can see this diagram. We can see in blue the common DNA sequences, the common nucleotides, the common genes between these species. And you'll see that a human and a chimpanzee are pretty much identical as far as everything goes. We have a very minor difference if I get my red pen out here and we circle this very minor difference between the human and chimpanzee genetic code, but everything else is much the same. When we then look at the gorilla and we compare it to, say, the human and the gorilla, and I'll do this, apologies, I'll do this in green here. We obviously have this band missing at the start for humans, however gorillas have this one present. When we compare to the chimpanzee and to the human, we can see that we have slight variation here as well. We're also missing this sequence in the gorilla, however the end is much the same. Then when we look down at the orangutan, we can see that there are significantly more differences compared to the other three species altogether. So when we talk about comparative genomics, the overarching definition of this is that we're talking about the comparison of the entire genomes of different species. Now the why, if you like, what is the purpose of all of this, is to help us identify similarities and differences in that DNA sequence or in that gene between different species. Now, we can do a whole bunch of different comparisons with comparative genomics. We can look at, as we've just mentioned, the DNA sequence. We can look at, as we've just mentioned, specific genes and the gene sequence. We can even look at the entire genome. Something that we haven't spoken about this year, but we will now, is we can also look at similarities and differences in the mitochondrial DNA. Now, all of these things together can provide us with evidence for evolution. And this is our key understanding in Year 12. This is what it all comes back to. So there's almost a script that I need you to learn, and it's this script down the bottom here, because it applies to all of these comparison points up here. So this is what we are comparing, so the similarities and differences of, and this is where we can apply our script. So the script pretty much remains the same, but long story short, fewer differences in insert thing here, so DNA sequence, specific genes, gene sequence, entire genome, mitochondrial DNA, so fill in the blank there, indicates a closer evolutionary relationship between species. This will mean a more common recent ancestor and less mutations accumulated. So if I get rid of this highlighting here and we change all of this right now to pink, the DNA sequence in here is the only thing that we need to change, really. We can keep the rest of this the same, okay? And so what I would be rote learning if I was you is simply this yellow script here so that you can include this in both your short and extended response answer questions. So what we have here is a quick comparison of some of the different genome sizes between humans and a bunch of other species. So you notice what we already know, humans have 46 chromosomes. What you may or may not have known is that they have 3 billion base pairs roughly, and an estimated gene number of 21,000. Now we can compare ourselves to a mouse who, sure, they have a different number of chromosomes, they have roughly the same number of genes, and there's quite a big difference there. I know it only seems small, the 0.1 billion. However, that is still quite a significant difference in terms of the number of base pairs. But as we go all the way down to something, I'll use my blue pen here, like bacteria with 4.6 million base pairs, only have one chromosome, 3,200 genes. So we can have great variation in terms of genome sizes. It just depends on what the genes are being used and potentially how evolved that species is. So this here is another representation. This is something known as a phylogenetic tree. Now, when we're talking about phylogenetic, I'm running out of space here, phylogenetic tree, these ones here allow us to determine the similarities between different species and identify points in which they have branched off. Now, the point with this particular phylogenetic tree is looking at these things known as ERVs, or endogenous retroviruses. Endogenous retroviruses are simply viruses that affected the gametes of a particular species at a particular point in time, and because they've affected the gametes, they are going to be found in every ancestor of that particular individual that was initially affected. So as we can see here, and the way that we can use endogenous retroviruses is, if we look at humans, humans currently have endogenous retrovirus 5, and they are the only species out of the great apes, the gibbons, the monkeys, and humans, or group of organisms anyway, with this endogenous retrovirus. Now, what this suggests to us is that humans were the only ones affected by endogenous retrovirus 5 because there had already been a branching point where they were no longer sharing a common ancestor with the great apes. So the ancestor that was affected by this did not progress down this great ape line. When we compare that now to endogenous retrovirus 4, on the other hand, you'll see that this is found in both great apes and in humans, and this suggests that it is slightly older, perhaps, this DNA sequence, and it's good evidence to suggest that, okay, humans and apes are not as common. So what this shows is an accumulation of genetic differences over time. And as we've said in our script up the top here, fewer differences indicates a closer evolutionary relationship. So if we have more differences in DNA, in the DNA sequence or in the genes found, this is going to indicate a more distant relationship between species. So let's have a look here at the ERVs themselves. So what are they? Again, as a virus, they're a viral sequence, and they've become part of an organism's genome. How does this work? Well, this is just simply with viral replication. The viral RNA itself is inserted from the virus into the host cell's DNA and is incorporated into it. Now, in terms of our evidence for evolution, this is where our script comes back into play. So fewer differences in this case, endogenous retrovirus locations indicates a closer evolutionary relationship between species, meaning a more recent common ancestor, and less mutations accumulated. So, organisms that have these ERVs in common with one another are more likely to have a more recent common ancestor. Those with less will have a more distant common ancestor. So humans have a more recent common ancestor with the great apes, say, as they have 3 ERVs in common, as opposed to humans with monkeys who only have 1 ERV in common. So as we have a look down, we're now on to our final kind of example of comparative genomics. We're looking at mitochondrial DNA. So historically, we've really focused on nuclear DNA, which is DNA found in the nucleus, double helix structure. We've got thousands of genes that control the function of the entire cell. And you get that nuclear DNA from not only mum, but also dad. The final thing to note with nuclear DNA is while it does mutate, the mutation rate is significantly lower than what we're going to talk about now, which is mitochondrial DNA, or otherwise known as mtDNA. So in terms of this mtDNA, where's it found? Well, the name gives it away. Mitochondrial DNA is found in the mitochondria only. Now, it's a small circular molecule, as we can see over on the right-hand side of the slide here. And this circular molecule itself is quite small, only containing 37 genes, as opposed to the thousands that are contained within the nucleus of nuclear DNA. From the function point of view, while nuclear DNA controls the functioning of the entire cell or the entire organism, mitochondrial DNA is very specific in its focus, and its 37 genes control the function of the mitochondria only, okay, that mitochondrial function. Now, uniquely, mitochondrial DNA is only inherited from the mother, and it has a very high mutation rate. And it's these final 2 points here, so put a star next to maternal and high rate of mutation, that allow it to be great evidence for evolution. Okay? So when we look at mitochondrial DNA, over time, it has been very slowly diverging from the mitochondrial DNA of the original female ancestor. Now, the amount of mutation or the difference in the mitochondrial DNA between 2 different individuals is going to indicate how closely related they are. So this comes back to our script once again. Fewer differences in this case, the DNA sequence, but what we're really talking about here, to be clear, in brackets, mtDNA, indicates a closer, indicates a closer evolutionary relationship between the species, a more recent common ancestor, and less mutations accumulated. So this here is a diagram just very quickly showing the inheritance of nuclear DNA over on the left-hand side. We can see that it's passed throughout the generations from both maternal and paternal partners. However, when we look at the mitochondrial DNA, it is only passed through the maternal line.